Welcome everybody to the final installment of the Neuro Light Lunch seminar series sponsored by Canadian Neurophotonics platform. Today, we're lucky to have two speakers that were originally scheduled to speak first in the satellite meeting for CAN, which was canceled, and then they were set to open our seminar series a month ago, and we had to reschedule because of the Black Lives Matter event at Neuroscience, SFN. And so I'm very happy that we're able to bring them back. So instead of opening on this high note, we're closing on this high note, which is great. So we'll have two speakers today, as usual. Our second speaker will be Alex Lohman, and I'll introduce him after. Our first speaker today is Mihaela Iordanova. Mihaela is an associate professor in the Department of Psychology at Concordia University. And she, in particular, has she's had a very good few years, for sure. She had a number of high-profile papers recently. She's also the 2020 CAN Young Investigator Award recipient. And so it would have been nice to hear her speak at CAN. So you'll get to hear her here. It's the next best thing. Mihaela is a Canada Research Chair. She did her PhD at the University of New South Wales and postdoctoral training in Cardiff, where she was just telling me about how wonderful the weather is, and at the NIH, before starting her independent position at Concordia in 2014. And so Mihaela has done really beautiful work combining sort of behavioral analysis of learning and the dopamine system using optogenetic approaches and imaging. And so with no further ado, I'd like to hand the platform over to Mihaela.
Thanks, Ed. So just let me know, can you see?
Yeah, it looks great. I would just add one more thing, which is for the audience, if you do have questions, please write them in the chat, and at the end of Mihaela's talk, we'll unmute you so you can ask your questions. Thanks.
Sorry, I just need to shrink this a little bit so I can see my slides. Okay, well, thanks, Ed, for this really nice introduction. And I also wanted to thank the whole Neurophotonics team for thinking of me, and as well as Sheena Jocelyn, so for thinking of me when they were putting together this series of talks. So what I'm going to talk about today is really a single study, or more like a two-part study that looks at the role of VTA dopamine transients in temporal-difference learning. And a big shout out to Etienne Maes, who is a very talented undergraduate student in the lab who collected all the data that I'm going to present today, and is moving on to the very prestigious combined MD PhD program at Miguel. But also to Alex Usipchak, who's a graduate student in the lab, and Megan Lozzi, who is also another talented undergraduate student. They were really instrumental in helping Etienne collect some of these data when we were going through the review process. And of course, our collaborators, Jeff Schoenbaum, who's fantastic, Mel Sharp, who's now at UCLA, and Matt Gardner. All right, so a lot of you would have heard about this link that's been established between the VTA dopamine signal and reward prediction error. And these data were first reported in a classical conditioning procedure where we have a cue that is paired with reward. So you can see this light here, a visual cue paired with reward. And what we see in terms of the dopamine signal is this elevation in firing rate to an unexpected reward. But as the reward becomes predicted, as a result of these pairings between the CS and the US, between the CS and the reward, this elevation in firing rate that we see at this point, at time of the surprising reward, is no longer present because this reward is no longer surprising, it's expected. Instead, what we see is a migration of the signal to the cue that predicts reward. And another important aspect of this signature is that if we were to present the CS but we don't deliver the reward. So we omit this expected reward. We see a depression in the firing rate. So in essence, what we have is this bidirectional signal, which reflects a positive prediction error at this time point and a negative prediction error here. So even though there's a, fairly convincing data in thinking about dopamine as a reward prediction error signal, the really quintessential design that looks at or is able to isolate prediction error in learning is blocking. So in blocking, what we have is two groups here, the blocking and the control group, and we have essentially two phases in this design. So this is the first phase, and this represents the second phase. So in the first phase, we present a cue, here a visual cue that's paired with reward, and we only do this for the blocking group. And once we have established this relationship between the visual cue and reward, we move on to the second phase. And in the second phase, we have an audiovisual compound for both groups, so an auditory cue together with this visual cue that was pre-trained in the blocking group, and we pair this audiovisual compound with reward. So everything in this second phase is identical between our two conditions, the blocking condition and the control condition. Equal number of pairings, the same cues, the same reward. And what we're interested to know is how much learning accrues to this auditory cue. How much is learned about the relationship between the auditory cue and reward? And generally, this is what we tend to see. So if we present the auditory cue non-reinforced on test, and we're looking for some sort of a behavioral readout about learning, so the conditional response could be something like magazine approach of the place where reward is normally delivered. What we see is that responding to the auditory cue in the blocking condition is much lower in comparison to the control condition. So this difference right there tells us that less was learned about the associative relationship between the auditory cue and reward in the blocking condition in comparison to the auditory cue and reward in the control condition. And before I go into why that is, I wanted to give you a real life example of blocking. A real life example that is quite specific to me, but maybe some of you can relate to it. So before moving to Canada, I lived in the States, and a very good predictor for the value of a dollar note in the US is the number that is printed on the dollar bill. So 5 or 10 or 50, 100. Didn't see a lot of hundreds, of course. And when I moved to Canada, what I noticed is that you could still use the number as a predictor for value, but also the notes are color-coded. Unfortunately for me, what happened was if you were to present me with the blue note, I would actually have no idea what the value of the blue note is. And essentially what happened is I had trained to use the number as a predictor for the value of the dollar bill, and I failed to learn the relationship between color and that value, which can actually be quite handy because if you have your money crumbled up in your pocket or in your wallet, it will be much easier to know what you're pulling out without having to search for that number. All right, so why does blocking happen? Why did the number block my ability to learn the relationship between color and the value of the dollars? Or why are the animals not learning the relationship between the auditory cue and reward in the blocking condition? Well, the reason for that is really prediction error. So here I have two equations that really capture this in a mathematical formula. At the top, we have the temporal-difference prediction error algorithm by Sutton and Bartow, and really in the bottom here is a modified version of the Roscoe-Wagner model. And both of these equations really tell us that learning is greater when you have a large error. So in the blocking condition, error is very small because we have pre-trained a cue to signal reward. So when the animals are presented with the auditory cue and the visual cue, and the audiovisual compound is fed with reward, they actually expect that reward. So they're not surprised by the reward. There's very little prediction error. The learning mechanism isn't engaged. And as a result, no learning or very limited learning takes place between the auditory cue and reward. And that doesn't happen in the control condition because the animals come in with no expectation of reward. So when the reward is delivered, they're surprised. This engages the learning mechanism and learning takes place. So what I have here is some other data from Schultz, where he's looking at the dopamine signature during blocking. And on the left, we had the blocking condition. On the right, we had the control condition. And we're looking at what happens to the firing of dopamine neurons when a reward is delivered after the AX compound, so after the blocking compound, in comparison to the BY compound, which is the control compound. So in essence, what we have is the same reward. Here, the delivery of that reward is surprising, and you can see that the firing of dopamine neurons is greater in comparison to firing to the same reward but following the blocking compound, right? So that reward right here is not surprising. And if we were to then look and see what happens in terms of the individual cues, so the blocking QX and the control QY, if no reward is delivered following X, we see that firing is at baseline. But if no reward is delivered following Y, we see this inhibition in the signal, which is indicative that the animals expected a reward and that expected reward was not delivered. So this is our negative prediction error. And if we were to actually present a reward following the presentation of X and following the presentation of Y, we only see this elevation in firing rate following X, because that reward is surprising, but not following Y, because they expected the delivery of reward. So in addition to these electrophysiological data, and there are really many more that have come since providing evidence for the role of dopamine in prediction error, we also have some causal evidence. And there are many studies here too, but I'm just really going to pull out one, which is the TH3 rat that's characterized by Ilana Witten. And here, Elizabeth Steinberg really conducted a very clever study where she used blocking, and what she decided to do was artificially boost the dopamine signal using channelrhodopsin at time of the expected reward in blocking. The idea here is if we were to boost that signal, then we should reinstate error. And in essence, we should encourage learning between the blocked cue, in this case it's visual cue, and the reward that is delivered. And we can see that here on test when we're looking at percent time spent in the port during the cue. This is our experimental group, and she saw higher level of responding to the visual stimulus in the experimental group in comparison to blocking control conditions. So providing causal evidence for the importance of dopamine in prediction error. All right, so the bottom line here is that there's been a lot of evidence that looks at the VTA dopamine signal at time of reward delivery, and that it seems to, and the data seem to suggest that it is acting as a reward prediction error. But we have very little out there that really looks at what happens to the cue-evoked dopamine signal. So what is that signal? What does that represent? And remember, in the first slide, I showed you that at time of reward, we have this dopamine signal when the reward is unexpected, and then the signal migrates to the CS. So in essence, we're asking two questions about this cue-evoked dopamine signal. Could it be a temporal difference prediction error that has migrated from the time of reward and now it moves to the cue? And in essence, what that means is that this signal should reinforce learning about that cue, or could it be a prediction about upcoming reward? So the good predictor essentially tells us that reward is coming. So could the dopamine signal represent this expectation of reward that is yet to come? So we set out to try and basically answer this question. And the way that we've decided to do this is using the TH3 rat and halorhodopsin, and we're going to mess with the signal at time of the CS. So this cue-evoked dopamine signal, we're going to attenuate it and see how that affects learning. So we firstly decided to model our two hypotheses. So our first hypothesis centers on dopamine transients corresponding to TD errors, and we used the Sutton-Barto algorithm, and we added another parameter. So this parameter here is nu, and nu essentially represents our attenuation of the cue-evoked dopamine signal. So we give nu a 0 when the laser is on and 1 when the laser is off. So when the laser is off, we're not modulating that signal at all. So the error here is going to be normal. So we've put nu outside of the error term, and error would be normal when the laser is off. and we would have a 1 value for mu. Now, if we are going to be attenuating that error signal, then we give mu a 0, and that's going to zero out the error at time of the cue. Now, the other hypothesis centers on dopamine transients corresponding to a prediction. And here we use the exact same algorithm, but if we want to modulate the prediction, we want to zero out the prediction, then our new parameter nu was inserted here, where the value, the state value is at each point in time, so at time t and t minus 1. The idea here is that we're not messing with the overall error. What we're doing is we're modulating the prediction, and that in turn can have effects on learning. So again, 0 is when our laser is on, so we would zero out these predictions here in our equation, and when the laser is off, we would have 1, so they remain normal. Okay, so the first question we're going to ask is the Q-evoked dopamine signal critical for learning? So to answer this, what we need to do is essentially use a design that looks at how learning takes place to an established predictor for reward. So here, what we use is second order conditioning. So in second order conditioning, again, we have two groups, a second order group and the control group. Again, we have a two-phase design. During the first phase of this experiment, we train a cue to signal reward, and we often call this cue a first order cue. So in both of our groups, what we have is we establish a good predictor for reward. Now, in the second phase of the experiment, what we're going to do is we're going to take another cue, an auditory cue here, a novel cue, and we're going to pair it with this good predictor in the second order group. But in the control group, we're going to present the exact same stimuli, and they're not paired. So the idea here is that this first order cue that has become a good predictor for reward should be able to reinforce learning about this novel stimulus. So essentially, what we have here is a second order conditional cue, but we should not have this in the control group because they're never directly paired, so they fail to be associated. So if we were to look at what we should see on test, when we present this auditory cue, and we're looking for this behavioral readout of the learning, so magazine approach, we should see higher level of responding to the second order cue in comparison to the control cue. Now, we can look at magazine approach. There's other ways to examine whether learning has taken place to an auditory cue. We could look at CS-based responding, such as orienting as well. All right, so this gives us a way to really prod the ability of a good predictor for reward and how it can reinforce learning. So what do we expect to happen to the dopamine signal during second order conditioning? So this is what this SOC stands for, second order conditioning. So here we have reward. When that reward is unexpected and presented, we're going to see an increase in the firing of dopamine neurons. Now, if we set up a first order cue, a good predictor for that reward, we're going to see the dopamine response migrate to this first order cue, the good predictor for reward. Now, in the second phase of our experiment, what we're going to do is we're going to pair an auditory cue with this good predictor, and we should see this dopamine response migrate to this earlier predictor, earlier reliable predictor for reward. And what's important to note here is that we're not presenting the outcome, right? So these pairings occur in the absence of reward, purely because this cue serves to reinforce learning about the auditory cue as a result of its prior pairings with the reward. So now we're going to use halo-rhodopsin to zero out this cue about dopamine, and essentially what that means is that we shouldn't see it back propagate to the second order cue. So if we're thinking about what we might see in terms of the conditioned response, then we really should see robust responding to this cue under normal conditions, but not when we're zeroing out this cue about dopamine. All right, so what we have here is our first experiment, which is looking at how does the cue about dopamine modulate second order conditioning. So in the conditioning phase, we present A+, so we set up A to be a good predictor for reward, and then we move into the second order training phase. So here we have two trial types. This is a within subject study. And we present QC, so C and D are auditory cues, and they're paired with A, the good predictor for reward. So essentially, C and D will both end up being second order cues, but these trial types are going to differ in terms of the neural manipulation that takes place. So we're going to attenuate the cue-invoked dopamine signal to A, so you can see that represented here, during the CA trial types, but not during the DA trial types. So we're going to do this during the intertrial interval. So in the middle here, we have our two models. So our dopamine is a TD error and dopamine is a prediction. And you can see here that if we were to attenuate the signal with our laser, then we should not see a cue-evoked dopamine signal to C. And what that means is that if we're looking at what should happen to the response, and this is our modeling, responding to C should be lower in comparison to D, right? And what I have further on the right is again our two models, but the data are presented in terms of differences in inhibition strength. So 0 is no inhibition, so responding to C and D would be equivalent. But as we increase the inhibition, and you can think about that in a number of ways, whether it's the amount of virus or how many neurons we end up attenuating the signal to, you can see that we'll consistently get the same kind of difference. So D, which is the purple, will be higher in comparison to C. So C will consistently be lower, and with increasing inhibition, we're going to see a greater difference between them. So let's have a look at the data. So these are the halorhodopsin animals, so responding to A as a result of conditioning is here. Then during second order training, what we have is equivalent responding to C, D, and A across the 2 days of training. And here on probe test, what you can see is that responding to C is lower, much lower in comparison to D. So this difference right there is in line with both of our models, which say that if you disrupt the cue-evoked dopamine signal, you're going to disrupt second order learning. And this is specific to having the opsin, because in the case of the EYFP controls that lack the opsin, you can see that responding to C and D is equivalent. So what this essentially shows is that if we mess with the cue-evoked dopamine signal, we mess with its ability to reinforce learning. But what it isn't telling us is, what is this signal? Is it a prediction or is it a TD error? So this is the second question that essentially we needed to answer. Is the cue-evoked dopamine signal a prediction about upcoming reward? So how are we going to go about answering this question? So here we end up using the blocking design. So like we have A, we train A, halfway through that training, we're going to present another stimulus, B minus, so we're going to set up a discrimination between these two. And then we're going to go into the critical learning phase, blocking phase. So during this phase, we have 3 different audiovisual compounds. Each of those compounds is paired with the same reward. BZ compound is our control compound because B is not good predictor for reward. So essentially we should have a fairly robust learning between Z and the reward. Now AX and AY are our 2 blocking compounds. A is a good predictor for reward, so it should block learning about X and the reward and Y and the reward. And the critical difference between these 2 compounds is that we're going to attenuate the cue-evoked dopamine signal to A on the AX trials, but not on the AY trials. And we want to look and see, does that affect learning about X and how does that compare to Y? Again, this is a within subjects design. So here again, we have models. So attenuating this cue-evoked signal is a TD error. As a TD error, will not do anything in terms of the response that we end up seeing of dopamine neurons to reward. And the idea is, if we are modulating the error, but we're leaving the prediction intact, then prediction comes in from somewhere else. And as a result, the animals will not be surprised by the delivery of reward. And you can see this in terms of what we expect for the conditioned response. Responding to Z should be high, that's our control cue, or higher anyway, in comparison to X and Y should be equivalent. And this again is our model where we look at different levels of inhibition strength, and we see no differences across these different levels of inhibition strength. If we look at our model where dopamine is a prediction about upcoming reward, then if this cue about dopamine gives us information about the reward that we are expecting, then if we were to attenuate it, what that means is that the animals will no longer predict reward, and that should reinstate the signal at time of reward. And remember from Elizabeth Steinberg's study that I mentioned a few slides ago, reinstating this dopamine signal at time of the reward in the blocking study will reinstate learning between X and the reward. So in essence, we should get unblocking. And you can see this here again in our modeling, where responding to Y, the normal block Q will be lower in comparison to Z. But if you look at responding to X, that should be quite high. So in essence, what this is unblocking. X should be higher than Y because learning has taken place between X and the reward. And the model here essentially shows the same thing. We can talk about these differences that occur at different levels of inhibition strength and why X would go beyond the control cue, for example, and why blocking would be even stronger to Y. We could deal with that in the question time. Okay, all right, so what do we see? So responding to A increases across days. When we look at A plus versus B minus, we have this discrimination between them by the end of training. In blocking, you can see that our two blocking compounds respond similarly, but our control compound starts slower, because they don't expect reward. But they end up at the same point by the end of training. So the moment of truth is what happens during the probe test. And here, what you can see is that responding to our control cue is high or higher in comparison to X and Y, but critically, X and Y don't differ from one another, which is in line with our dopamine as a TD error prediction and not with our dopamine as a prediction hypothesis. So these are the halorhodopsin animals. So if I was to show you the data for our control animals, so the EYFP animals, you can see, so this cell proceeds as normal, and you can see that X and Y, again, don't differ from one another. So our data are quite similar. All right, so basically, what I've shown you is that attenuating the VTA dopamine signal at time of a good predictor for reward disrupts that predictor's ability to reinforce further learning. So I showed you this in the second order design, where we disrupted second order learning, whereas this disruption is due to attenuating the prediction error signal carried by the cue, and not because we're disrupting the prediction about upcoming reward. And this is evident in the blocking study. So one thing I would have liked to do next, but I'm not able to really because of COVID, is tell you a little bit more about the kind of work that we've been doing in trying to understand the content of learning that is supported by the VTA dopamine signal. So second order learning is not really equivalent to first order learning, in the sense that in first order learning, an association forms between the neutral cue, initial neutral cue, the visual cue, and reward. And in second order learning, we don't have a reward that is delivered, but learning could occur between the auditory cue and the visual cue. But behavioral data have shown that this is not generally what happens in second order learning. In fact, an association doesn't seem to be between the auditory cue and the visual cue, does not seem to regulate responding during the second order cue. And we know this from some studies done by Peter Holland, where we can extinguish the first order association. So the visual cue, we can present it no reinforced and see a dramatic decline in responding to that cue. So essentially, in a way, breaking the association between the first order cue and reward. But what is really interesting here is that this extinction of the first order cue does not relate back to responding to the second order cue. So something else, it's not this association between the auditory cue and the visual cue that supports responding that we see in second order conditioning. And what will be really interesting to see is whether if we were to boost the dopamine signal, not only whether we can increase, augment second order learning, but more importantly, can we change the learning that takes place in second order conditioning? does this artificial stimulation of the dopamine signal lead to an association between the auditory cue and the visual cue? And there's some precedent to think that this could happen on the basis of data from Jeff Schoenbaum and Mel Sharp, who have shown that indeed stimulating Th positive cells in the Th3 rat can lead to learning between two neutral stimuli under conditions when normally this doesn't happen. But unfortunately, we'll have to stay tuned to find out whether that is the case. And so in addition to this, we're also looking at whether the signal regulates aversive prediction error. So just to end, like people did last week, our lab is hiring. So if you're interested in coming to Montreal, then get in touch. Thank you.
All right, thank you so much, Mihaela. That was great, really interesting. So if anyone has questions from the audience, please type them into the group chat, and we can activate your vignette. So I actually had a sort of a strange question that dopamine is clearly the main driver of this phenomenon, but are there other neuromodulators that play a similar role? Has that been examined to a certain extent?
Yeah, so that's a good question. So you think like in second order conditioning, is that what you're thinking? Or
exactly,
blocking,
yes.
or, yeah. Yeah, so yeah, for sure. I mean, Peter Holland has done some work looking in a very different place, looking at the amygdala and how that regulates second order conditioning. So absolutely, a postdoc in the lab, Valine DeLay, has also done that in fear, actually, and she's also shown some evidence for the amygdala in regulating that. We've also done some other work in fear, looking at the role of the orbitofrontal cortex in second order learning. And we're actually finding some really neat data. You know, I was kind of in two minds whether to present that or this, but we're finding a role for the OFC that is opposite between second order conditioning and basically the same task, where you reverse the two phases. We call it sensory preconditioning. So I'm certainly not suggesting that dopamine is the only one that regulates, you know, learning, not at all. But we can also modulate learning in a number of ways. And I guess the modeling kind of makes that really, really clear when we think about, if we mess with the error signal, we're going to disrupt second order conditioning. But we can also mess with the error signal indirectly through messing with the prediction, right? And the prediction could be somewhere else in a completely different place. And if we mess with that, then again, we can modulate or disrupt the second order conditioning phenomenon as well. So, you know, it's generally the way that I think about these things is that it really isn't happening in one place. And we've just kind of tried to elucidate using these behavioral designs what happens at the level of dopamine, but not necessarily what is the neural substrate that determines second order conditioning. I don't know if I'm being
no, that
clear
makes
about that
perfect
distinction.
sense, actually.
And you know, in blocking too, you know, there's other brain areas that have been found to be involved, not just dopamine in the VTA, but also in other areas like the accumbens or the amygdala. So, you know, learning happens everywhere, I guess.
All right, thanks. That's great. Actually, I knew Paul would have a question. There's a question from Paul Clark. Would you like to ask it yourself, Paul? Or?
Yeah, let's see your face, Paul.
You should be able to see my face.
Perfect.
Thanks, that was a great talk. So my question has to do with what exactly blocking is. So let's say you set it up so that the light perfectly predicts reward, and then you add a tone to the light. Now, I think you're saying that really very little is learned about the tone predicting the light, because it is redundant.
Sorry, the tone predicting the reward.
Yeah, sorry, the reward, yeah, sorry. Because it's redundant. However, what happens if you, let's say, extinguish the response to the light? Maybe you did learn something about the tone, but you weren't expressing it behaviorally.
Yeah, okay. So what you're getting at is, if we go through the normal blocking procedure, we set up the good predictor, the light, and then we're doing our blocking with the auditory and visual stimulus paired with light. And then instead of testing to the auditory cue, we extinguish the light and we look and see, hey, what happens to the auditory cue. So this is actually, it's something that was done by Ralph Miller. He has a very different way of thinking about learning. You know, his argument is we learn about all these relationships and how we express them is in terms of performance, right? So they're just essentially dormant, and depending on what you know, you make these comparisons between different associations, and they're going to determine the response that we see at the end. So his argument is normal learning takes place between the auditory cue and the reward, but you just don't express it. And his argument would be exactly what you say, that if you extinguish the light, then you should see responding to the auditory cue. And he has found that, but other people report that that isn't the case, and it's indeed a failure to learn the association between the auditory cue and the reward that is delivered. Now, one other thing that I want to mention here, though, is that, and there's other ways to kind of look at this. So there's another really clever study that was done where you could take that auditory cue that supposedly was blocked, and you could use it as a blocker for further learning. And if learning had taken place, whether it's expressed or not in performance, it should be able to block learning, and it doesn't, actually. But just to kind of clarify more generally, what happens in blocking is, you know, very often we talk about no learning taking place between the auditory cue and shock. That really isn't true, and you can see that in a lot of our behavioral data, that some responding does occur to that auditory cue. It's just not as much as it is to the control. And there are a number of ways to think about this. One of those, which is supported by theory, is that the first time you see that auditory cue, you attend to it because it's novel and you are going to learn about it. But what is going to happen is you're going to realize that it isn't as good a predictor about the reward as the other cue, and therefore you stop learning about it. So if I go back to, you know, the money example, I mean, I have noticed that Canadian money is, you know, color-coded. I haven't ignored that, right? It's just that I haven't formed the association because I have been focusing, attending to the number that is printed on the money, and I have used that to guide my behavioral response. So yeah, and you can encourage learning about blocking in other ways, which you probably know.
Great.
Thank you very much.
Yeah, thanks. So just because of time, there is a question from Tim Leung, which I would ask maybe you guys could take offline, or after the next speaker, you guys can.
I'll hang around for sure.
Excellent. Thank you so much. Please join me in thanking Mihaela. Fantastic talk. Applause is so sad on Zoom. That's the one thing we need to improve. We need an applause track.
So I'll stop sharing. And is Alex able to share, or am I still doing it?
Alex, are you in?
Yeah, I'm here. Can you hear me?
Yep.
I should be on.
I'll quickly introduce Alex. Alex is our next speaker. So Alex is at the Hotchkiss Brain Institute, University of Calgary. And he did his PhD work at the University of Virginia with Brant Isaacson, and his postdoctoral work at Calgary with Roger Thompson, where I think he started some of the projects that he'll be talking about today, looking at connection channels in the injured brain. Very exciting work and impactful. Roger is an assistant professor at the Cell Biology and Anatomy Department, Calgary, at the Hotchkiss Brain Institute. And Alex, take it away.
Perfect. Well, thank you so much for organizing this symposium online. It was really, great for us to be able to continue with the presentations in lieu of the cancellation of CAN. So yes, so you guys can see this, right? So as I've mentioned, I don't really need to go through this, but I'm not familiar with many of you on the feed, so I figured I'd just give you a brief background, kind of where I got to where I'm at now. So yeah, I did my bachelor's degree at the University of Missouri, graduated in 2010, working mostly on protein biochemistry and working with Bill Fay, and Steve Siegel in blood vessel control of blood clotting and blood pressure regulation as an undergrad. And that spurred me to go work for Dr. Brian Isaacson at the University of Virginia, who was really gung-ho looking at gap junctions and heterocellular communication in blood vessels themselves. And this really got me kind of on the pannexin forefront, which I'll talk to you mostly about today in terms of our tool development. But through my work there, looking at vascular inflammation, I met Roger at the annual Gap Junction conference, and knew that I needed to come to Calgary to really dive into neuroscience and the brain, and begin, contributing towards the pannexin, work that's happening in the brain. And I'll give you some of the motivation behind that as well. So my primary research interests in the lab are heterocellular communication between vascular cells, glia, peripheral immune cells, and neurons in both contexts of health and disease. And what we're really trying to accomplish is understand what are some key molecular signaling mechanisms that control the way these different cell types communicate with each other, and how are these mechanisms specific to whether inflammation in the brain is good or bad. And, the majority of the side of the work that I'm going to talk to you today is really focused on, based on the information that we discover from our molecular signaling kind of studies, can we then harness that to create new, more selective tools for optogenetic manipulation or sensing of different types of activity related to inflammation. So along these lines, we know that inflammation can be good and bad in the brain. For instance, microglia play central roles in regulating tissue homeostasis. They're involved in synaptic remodeling during neurodevelopment at different stages. They support neuronal health, but in response to injury or disease, they can become activated and adopt, various phenotypes along an inflammatory spectrum that contribute both good and bad things in the brain. So really, what are some of these central balancing acts that regulate this? And can we then harness that information for, therapeutic benefits when it comes to brain injury or other neurological diseases? So we also know that, we initially thought the brain was more of an immune privileged organ due to exclusion of dyes that were injected into the bloodstream did not seem to get into the CNS, and also the absence of classical antigen presenting cells, low levels of major histocompatibility, complex proteins, and the presence of a blood-brain barrier. In addition, the lymphatic side, where we see immune cells typically draining from tissues, seem to be missing in the brain. But we know now that this isn't completely the case. Seminal work identifying meningeal lymphatic vessels that has since been expanded is really contributing a lot to our understanding of how immune cells access the brain and how their routes of transit and communication are impacted. In addition, we know that in many neurological disorders and injuries that the blood-brain barrier breaks down, and we actually see infiltration of peripheral immune cells that are also contributing both beneficial and detrimental effects to neurological health. My focus in the neuroscience field has really been on the brain injury side of things. So during my postdoc with Roger, we focused mostly on ischemic stroke and roles for inflammation in the brain following stroke. And since opening my lab about roughly 2 years ago to the date, I've transitioned more into research surrounding traumatic brain injury, and specifically repetitive mild traumatic brain injuries during adolescent neurodevelopment. So one of the main axes that we're focused on, and really the motivation for investigating pannexin channels, is the role of purinergic signaling in regulating inflammation both in the periphery and the brain. So purinergic signaling is kind of a brilliant mechanism that cells have evolved to utilize an abundant energy source in the cell, ATP, and a plethora of enzymes that metabolize ATP to ADP, AMP, and adenosine, like the CD39, CD73 ectonucleotidases, as well as a gamut of purinergic receptors. I think today we're looking around 22 or 23 different classes or isoforms of both adenosine sensitive P1 type receptors and the ATP, ADP sensitive P2 type receptors. So this allows the cell to communicate in very sophisticated ways. And it turns out that purinergic signaling really is at the center of many of the immune responses throughout the body. So here I just wanted to highlight, that everything from monocytes to microglia to vascular endothelial cells differentially respond to ATP and its metabolites to regulate different aspects of inflammation, like cell migration, activation, phenotypic differentiation, as well as synthesis and release of signaling molecules involved in inflammatory signaling. Specifically towards my interest throughout my research career, I've been really focused on how purinergic signaling regulates the way immune cells in the periphery cross blood vessels into inflamed organs. And it turns out that purinergic signaling is really involved in multiple different aspects of how circulating immune cells are captured, how they roll, and how they migrate into these tissues. But really, what hasn't been known, while this is, purinergic signaling is really, has been around and been studied to a great extent in the context of inflammation, but how ATP and its metabolites are released from cells in a regulated fashion has really been the missing component to how diseases and injury cause this sort of response. So some of my work during my PhD was focused on these channels called pannexin-1 that emerged as prominent regulators of ATP release. And my graduate degree culminated in this study where we identified a signaling mechanism between pro-inflammatory cytokines like TNF-alpha and the activation of pannexin channels releasing ATP that's important for both the recruitment, adhesion, and migration of circulating leukocytes across the blood vessel wall. So in the brain, however, little's really been studied in terms of inflammation and pannexin signaling. And we've hypothesized that perhaps these channels sit at a key regulatory axis by either regulating access of peripheral immune cells to the brain, their activated phenotypes when they get into the brain, or other aspects of inflammation, and could therefore potentially provide a key target to modulate to improve or tip the scales more in the direction of protection versus destruction. So what are the pannexins? They were first identified in early 2000 through a homology screen of the invertebrate annexin gap junction proteins. They were found to have sequence homology to the annexins, but not to the eukaryotic gap junction proteins, the connexins. There's 3 isoforms that have been identified to date, pannexin 1, 2, and 3, and most of the work's focused on pannexin 1. This is probably because it's found to be fairly ubiquitously expressed throughout different cells and tissues in the body, but also the tools to study pannexin 1 and its prominence in identified ATP release pathways have really set the stage there. One of the initial work looking at pannexin suggested that pannexin 1 was a hexamer, and actually very recently with the advances in cryo-EM and high resolution structural analysis, we now know that it actually forms a heptamer, which now all of these diagrams and schematics seem to be wrong and will need to change moving forward. So focus mainly on pannexin-1. They're glycoproteins. They're glycosylated on their extracellular loops, which is involved in membrane trafficking. It also is thought to block gap junction formation between cells. So they actually exist as single membrane channels that permits access of things like ATP. Some ions tend to be anion selective, it appears, at least in the physiological sense. Small metabolites and fluorescent dyes between the intracellular and extracellular space. And I just wanted to highlight some of the regulation that we are aware of in terms of how the channel activates and closes, has focused on the C-terminal domain, the intracellular C-terminal tail that seems to be highly dynamic and acts in a sort of ball and chain mechanism. However, we know that it's not quite this simple. With the new heptameric crystal structures that are coming out, there are a few different routes for ion flux and metabolite flux through the channel, and it seems that the C-terminal domain is highly dynamic and regulating these things. So just a little bit on kind of what we know to date about how they're activated. Work from Roger's lab has really highlighted ischemia in the brain and opening pannexin channels. Caspase dependent cleavage of that C-terminal domain during apoptosis leads to a large pore formation and efflux of ATP. And you can see there are a number of different receptor dependent and independent mechanisms that have been suggested to regulate this. So the focus of the main data I want to show you today is how can we make better tools to investigate pannexin function. And this really stems from a lack of specific pharmacological agents, as well as reductions in, genetic compensation that often occurs in some of the transgenic mouse lines to knock out these proteins, while we're developing more specific inhibitors and mouse line strategies to avoid these issues. Where when I joined Roger's lab, we really wanted to try to harness optogenetics to find a more specific or selective way to investigate how these channels work. So let's see if you can see this. This is really fueled by trying to develop a protein with these characteristics in mind. So be selective, the illumination and the activity would be selective to your protein of interest, in this case pannexin-1. It would have similar activity characteristics as the endogenous protein in terms of what we know. It would really like high temporal regulation, you know, to the point where we can turn these things on and off in very sophisticated ways, and the ability to express and manipulate them in cell type specific manners as well. So when I was in Roger's lab, we formed a collaboration with Dr. Robert Campbell and one of his very talented PhD students, Wei Zhang, and sought to utilize this caspase cleavage mechanism to begin developing a light-regulated pannexin channel. This was stemmed from a new technology that Wei and Dr. Campbell developed based on their photoconvertible fluorescent protein M-maple. And maple is a green fluorescent protein that when you expose to a roughly 400 nanometer wavelength lights can photo switch its chromophore into a red shifted state. And this occurs through a beta elimination reaction in the central chromophore that's residing in the pore of these beta barrel fluorescent protein structures. So Dr. Campbell's group was able to use, you know, rational design where you could reposition the N and the C termini in relation to where that peptide backbone break occurs in the chromophore, and possibly allow for release of basically a split chromophore where you could then tag the N and C termini with different proteins of interest to have a sort of photo-uncaging effect. So this is just kind of showing the basics of their focal, the new cleavable protein. In the green states, in unstimulated, it is a green fluorescent protein. However, with 380 to 400 nanometer light, you get a transient shift to that red states when you have the beta elimination reaction in the chromophore. And this can then spontaneously dissociate to release the C-terminal N that's in proximity to where that break occurs. And this results in a loss of fluorescence. So you can use loss of green fluorescence as an output for the extent of photoconversion that you've had here. So in order to target pannexin in a specific way and regulate this protein, we needed to have a system where we could selectively cleave that C-terminal domain that seems to be auto-regulatory in the way the channel opens. So Dr. Campbell's group worked on developing a novel light-controlled protease from the hepatitis C virus, NS34A, I believe. But essentially what they were able to accomplish was taking the catalytic domain of this viral protease and tethering an inhibitory peptide into the active site through the photocleavable protein. In doing so, they tested a number of different inhibitory peptides for this protease and settled on this CP5, 46A, 4D5E peptide here, which had a strong enough KI to where you would have minimal basal activity from the protease itself, but will not bind so strong that when you photocleave the focal protein that you will never get dissociation. So we call this focal HCB protease. This is a photo-uncaged protease that we could then go into the pannexin-1 protein itself and mutagenize the caspase cleavage domain to a site that would be specifically recognized by this viral protease. So when we illuminate with violet light in the 380 to 400 nanometer range, we should get release of this inhibitory peptide, revealing an active protease that then can selectively target the sequence we've engineered into this autoregulatory domain of the pannexin channel. So we did a series of in vitro screening and optimizations for what we're now calling optopannexin-1, which is this two protein system. We're able to use small molecular weight fluorescent dyes to look at dye uptake through these channels. In the top panel, there we go. Can you guys see my laser pointer? There we go. In the top panel here, you can see just the expression of both focal and green and an M-cherry tagged pannexin channel that we modified that cleavage site. And when we stimulate with violet light, we see this reduction in green fluorescence, indicating that the protein has been, that the photocleavable protein focal has cleaved and dissociated. And we see a concomitant time-dependent increase in TOPR03 uptake into these cells. And as a nice control for all these studies, we used a version of the protease with the M-maple version, which cannot dissociate, and we do not see any dye uptake into these cells either. As kind of more selective outputs that we're interested in looking at ATP release, we're also able to use photoconversion of our optopannexin-1 system to drive ATP release from cultured cells, similar to functions that pannexin has both in the physiological and pathological sense. We also performed some patch clamp electrophysiology. There's very minimal residual current from pannexin channels that have their C-terminal tail. These are voltage ramps and HEK cells that we've co-expressed the optopannexin-1 constructs. When we photoconvert, we get a large increase in this hourly rectifying current, and we can block this with this pharmacological agent carbinoxolone, which is often used in these reduced in vitro systems as a pannexin blocker. However, it is also semi-dirty in that it can block gap junctions and connexins. And here's just an example of a time course of the electrical current that we get through these channels. So as a proof of principle that this construct can be used to mimic connexin function that's been established in vivo, I went ahead to assess whether or not we could use optoconnexin 1 to drive excitotoxicity in the context of ischemic stroke. So work from Roger's lab, both from his postdoc and some of the studies we did with Dr. Nick Willinger before he moved to UBC, identified connexin channels and excitatory neurons as a terminal channel that leads to the influx of calcium and cell death that occurs in response to glutamate toxicity. So we were able to package the photocleavable protease and the modified connexin in a bicistronic vector for co-expression under a LOX-P system to get cell type specific selectivity. And we used primary neurons and brain slices from CAMK2 Cree rats to virally transduce these things. So here, the main thing to look at is in wild type primary rat hippocampal neurons, we have DS red fluorescence. We don't see any green fluorescence, so this is turned off due to this premature stop codon. And in our CAMK2 Cree primary hippocampal neurons, we now see expression of our green focal protein and presumably connexin 1. So from work in Roger's lab by Dr. Willinger, they developed a model using NMDA to simulate glutamate toxicity. And the key hallmarks here are dendritic blebbing on these excitatory neurons, as well as the formation of these large inward currents. And what we could do is photostimulate connexin in the absence of NMDA overstimulation or excitotoxic concentrations of the NMDA receptor agonists, and cause dendritic blebbing and these large inward currents in the absence of that, suggesting that connexin 1 is really sufficient for driving those effects. So just in the last couple of slides here, I want to tell you about some things we're doing to try to expand these tools for further use. So the original optoconnexin channel was able to be fairly selective for the protein of interest, in that our viral protease only detects the engineered sequence that we put into the modified connexin. It seems to release ATP and have electrical characteristics similar to endogenous connexin. However, we really don't have high temporal regulation. Once you photocleave the protease, releasing it to cleave the connexin C-tails, there's really no way to turn it off. So one thing that I have been working on at the end of my postdoc in Roger's lab is to try to develop a different version that would allow for temporal light on off kinetics for opening the channel. And this is based on this photo actuator called ASLove2. It's from a phototropin called Avena sativa, and it's the light oxygen voltage sensing domain. This is an anti-parallel 5-stranded beta sheet that has this structured alpha helical domain called the J alpha helix that sandwiches a flavin mononucleotide cofactor. And upon stimulation with 450 nanometer light, you get the formation of a new covalent bond between a cysteine and the beta sheet and the FMN molecule, leading to unfolding of this J alpha helix, which can be used as an actuator or a mechanical arm to control conformational changes. And this is just proof of concept here, where Anna Moroni's group and colleagues engineered the first light-gated potassium channel using the LOVE2 domain, where they were able to attach the LOVE domain to a mechanically sensitive domain in this viral potassium channel and achieve blue light stimulated inward currents due to unfolding of that J alpha helix and the associated conformational changes. So what I decided to do was to try to incorporate that LOVE2 domain into the C-terminal domain of pannexin-1 in an attempt to use the actuator activity of that J-alpha helix unfolding to remove the C-terminal autoregulatory domain from the conductance pore. And I essentially went in with the LOVE2 domain and made a series of chimeras starting right near the beginning of the intracellular C-terminal domain, all the way up to the caspase cleavage site at every third amino acid to develop a screen. So first things first, we checked for membrane localization of our modified chimeras using biotinylation. So here you can see all of these constructs at these amino acid positions traffic to the membrane. We confirmed this with some immunofluorescence as well, and it seemed like in culture we were getting decent membrane expression. And then I went on to perform dye uptake assays as more of a crude screening approach. And what we found was if we inserted the LOVE domain around this area between 369 and 375, this is amino acid number in the pannexin monomer, we could get blue light induced uptake of dye. And whether or not it closes or not, we did this follow-up experiment where we stimulated with blue lights and then measured the uptake of topro dye for essentially 5-minute periods, and then stimulated again to see if we could get further uptake, with the assumption that if the channel was staying open, this should be a pretty linear curve here, but it looks like it's likely closing to some extent as we're not seeing massive influx there. Finally, the electrophysiological characterization. This is the 372 chimera with the LOVE domain and pannexin-1 looking at blue light induced currents, and we were able to find some cells where we could get blue light induced pannexin activity that then would return to baseline in the dark state. However, we have pretty low N values right now, and not all of the cells responded this nicely. So we're working on trying to move forward with this and refine to where it's a little more of a steady, controllable protein. It may have a lot to do with the fact that the new crystal structure crystal structures that have come out for pannexin-1 show a few different conductance pathways. So when the C-tail is in the pore, there appears to be this vestibule on the side of the protein on the intracellular domain where you can still get ion flux. So this is a very complicated channel in that sense, and understanding more and harnessing the information from the structural data that's coming out now will help us refine this. If I have just another minute or so, I just wanted to briefly show what we've been doing recently in the lab, still related to pannexin-1, and what our NSERC is funded on, is to develop tools to study inflammatory cytokine signaling. And in relation to pannexin-1, there's a plethora of studies that have come out showing that interactions of pannexin-1 with the P2X7 receptor regulate assembly of the inflammasome and production and release of pro-inflammatory cytokines like interleukin-1 beta and IL-18. So we thought first we could develop a system where we could selectively process interleukin-1 beta and release it from cells to have an optically controlled system to do a cell type selective release of interleukin-1 beta in vivo. And the way we approach this is very similar to the pannexin, optopannexin, in that the pro-IL-1 beta protein has 2 caspase domains, cleavage domains in this pro domain that need to be cleaved in order to form mature IL-1 beta that can then be functionally and biologically active. And we were approaching this by mutating these caspase-1 cleavage sites to those that could be recognized by the protease, where we could then do photoactivation of this focal HCV protease and cause selective processing of pro-IL-1 beta into the mature form. this work is done by an undergraduate student in the lab, Abigail Treblecock, and it's still very preliminary, but you can see in cells co-transfected with these constructs, we get nice green fluorescence indicating our photocleavable protease. We photoconvert, we get some loss of that green fluorescence indicating that we're getting cleavage and activation of the protease, and then we get transient red fluorescence, again, due to the nature of the way focal converts to the dissociable protein. And then we harvested whole cell lysates and looked for conversion of pro-IL-1 beta, or modified pro-IL-1 beta, into a mature form. So this is still work in progress, but you can see this band here is the pro full-length IL-1 beta, and down here we have mature interleukin-1 beta, which has then cleaved off the prodomains. And right now what we can say is that when we don't have our protease present, we don't really see much of that maturation. However, when we do have the protease co-transfected, we are getting processing. Right now, the hitch is we seem to have some basal activity of this protease where even under no light stimulation, we're getting conversion. So we're working on using a couple different forms of this. The last thing we're doing, and I don't have any data for this yet, is to try to make an interleukin-1 beta biosensor system using a FRET-based approach. And the rationale behind this is that, you know, analysis of pro-inflammatory cytokines in the brain typically requires you to extract the brain and grind up the tissue and run ELISAs, or do some sort of immunostaining on fixed sections, which loses both spatial and temporal selectivity. So the neat thing about the interleukin-1 receptor type 1 is that when interleukin-1 beta binds to it, it recruits an accessory protein to form this dimeric heterotrimeric complex with the interleukin-1 beta, the IL-1 receptor 1, and the accessory protein that leads to signaling. So based on crystallographic structural data of IL-1 receptor and its accessory protein, the C-terminal intracellular domains appear to be in very close proximity to each other, and we are now developing constructs to attach FRET pairs and begin testing whether we can get a FRET signal through IL-1 binding that can later be refined and incorporated into viral delivery strategies to get you know, real-time in vivo FRET-FLIN analysis of this specific cytokine. So that's all I had prepared today. Sorry I went a little long there. Just like to thank the members of my lab. Abigail was responsible for the photocleavable IL-1 beta work that's ongoing. My research associate, Dr. Robin Flynn, is really pioneering a lot of the protein engineering things in the lab and is focused right now on the IL-1 biosensor. Eric Yolfson and Thomas Carr are PhD students in the lab who are more working right now on the TBI-centric projects. And Asher and Andy are undergraduates in the lab as well who are contributing basically across the board. Obviously need to thank Roger Thompson for training me for those 4 years before becoming independent. And Ali Werner, right now a PhD student in Roger's lab, has been working on the Pennexin Love stuff moving forward. And obviously none of this work's possible without Robert Campbell and Wei Zhang, who did a ton of work in developing the photocleavable protein and the associated protease with that. So with that, I'd like to take any questions and obviously thank my funding support.
Thanks a lot, Alex. That was great. So if you have any questions for Alex, please type them into the chat. I actually had a question. So I'm pretty excited about the IL-1 FRET reporter, the receptor reporter. Do you think that the overexpression of the receptor would in some way cause spontaneous activation, or are there issues there that you're concerned about?
Yeah, so I didn't really mention it, but what we're working with right now is a couple of different approaches. Obviously, the IL-1 receptor type 1 is the major one with the crystallographic structural data. But the type 2 receptor actually lacks its intracellular catalytic domains and acts as a decoy receptor to modulate extracellular IL-1 beta signaling. So potentially using that construct versus the active signal transducing type could help prevent maybe basal, overactivation of IL-1 signaling pathways. However, obviously titrating these things, particularly when you're taking viral approaches to express these in vivo and figuring out the right titration so that you don't have an overexpression artifact is important. So. Lots to do there still, but.
No, thanks. That's great. Any other questions? I guess we've run a little bit late, so I don't see any questions in the chat group. If that's the case, please join me to thank both of our speakers today, Mihaela and Alex. It's really great to have you guys here to close the session. And thanks to everybody in the audience who came to participate over the last month. And with that, I guess I'll close out the session. You guys can continue to talk offline. Thanks again for coming. See you again at Cannes next year, hopefully.
So I don't know if Tim is still here, but I could answer his question.
Is Tim here?
It was,
do you want to unmute Tim?
It doesn't really matter anyway.
He is here, yeah.
Yeah. So basically he's asking whether, the optogenetic inhibition in our studies could have effects beyond depressing the prediction error signal, the temporal difference prediction error signal. So this is a really important question. So could it be, say, punishing behavior in a way? So like when we, when we pair C with A and then we're attenuating this dopamine signal at the beginning of A, the animal should be going to the magazine when A is presented. So if indeed we are punishing that behavior, then that could disrupt the transfer of conditioning to C. And a really easy way to look for this is to look at the conditioned response to A when you have inhibition of the dopamine signal, or at least attenuation of the dopamine signal, versus A in the other trial types when that attenuation of the signal happens during the ATI. And we looked at this early in the queue and later in the queue, and basically we see no differences. So there's no reason to think that, that's what dopamine could be doing. But, it's an important point because we do have studies looking at the role of a dopamine transient in aversive prediction error, and we are finding some evidence for that in the opposite way to the way that it works in reward. So it's kind of important to always check these things.
If there's nothing else, thanks again, guys. I really appreciate you offering to speak. It was great to hear about what you're doing. I can't believe you're just down the street and we haven't encountered each other yet. So
yeah.
Hopefully we'll have more opportunities in the future.
Yeah, absolutely. That'd be great.
Good luck to both of you.
Thanks so much.
Yes, Ed. Yeah, that was awesome.
Exciting stuff going on. I actually, at some point, I'll probably get in touch with you about trying out some of those tools. It's really exciting. Once you're ready to
we want to share. So obviously, the point of making them is so they're useful for the community. And yeah, we're not quite on the level of the Campbell lab, but we're trying to take a different approach in the way we're thinking about specific proteins of interest and try to use some of the existing tech that's out there in a new way to bucks. So
that's a pretty high standard. Yeah. All right, excellent. It's great to hear about everything. I'm going to close the session then. See you guys at Cannes, hopefully. Bye.
Take care. Bye.